based on the barrier, you see how well I did. What means you fill up the blanks. Right? So that's the bottom line. So remember that one more time. You always can write a free energy as a function of any constraint you want. Nobody can forbid you to do that. That's mathematically guaranteed. As long as you do enough sampling, that's fine. Now, that f of q, or whatever variable you have, is a potential of mean force in a sense you can talk about. That barrier has some meaning, and you're actually moving on that effective potential. And you've got a problem of many, many dimensions, protein folding reduced to a few dimensions. Is that a real decent kinetic coordinate? That's another story. Okay? And works in some cases and don't work in the other case. So remember the name of these words. They all mixed all the time. Oh, free energy function, QPMF. No, but remember that base. Free energy is always allowed to do that. You can always write the tree. But the question is that something that's kinetic value? Now, I'm going to give a little bit of an introduction for something that Cecilia did several years ago when she joined us in the group. I just want to bring this up just out of curiosity today, to bring a little bit of how you go to off lattice. I won't have much time to do that today because I'm running out of time. At least I did what I want to do today. Is the following situation. As I showed you on the last slide, what I showed you, there are two things that happens. And I'm going to put the words. I put these words on the board. You know, I told you energetic frustration and topological frustration. I said this is a meaningless word, just come as a parallel to this one, to tell you the geometrical difficulty into folding. Now, if you look at several experimental papers, they're going to tell you proteins folding is controlled by topology. You're going to find lots of papers saying that. That's a very stupid statement. Okay, let me tell you exactly what's true. What that means, and in practice that's what you observe. You say, what do you mean you observe? You just show me a slide that tells me the heterogeneity of which contacts are formed first and later depends on geometry. Now you're telling me this is a stupid statement. What I'm telling you is that proteins, real proteins, and that's what the last slide showed you, have done a sufficiently good job on the energetic aspect of the problem. Means they have reduced energetic frustrations. Means they have made TF over TG 1.6 or larger. There's big arguments what this number is for real proteins. I think the number around 2. In practice, there's some people say it's even 4. But the bottom line is not 1.3. 1.3 is too small. Means if TF over TG is large, you dealt well with the energetic frustration part of the protein. Then what's left is the topology. This is the only final difficulty. But doesn't mean that that's the main difficulty. That means that someone took the major difficulty out of the problem, then you're left with the smaller one. So the fact that folding, you can explain your folding in terms of geometry. And if you go out there on the current folding field today, some people use this approach without even noticing. So when you have a big thing that makes Baker and Black's things like contact order, that's something that just comes from the native structure of the protein. The fact you can make comments and conclusions just based on the native structures tells you that energetic frustration is not a big problem. Because you don't know what contacts are formed but on the unfolded basis by just looking at the native base, right? So the fact that you can comment on the native base tells you that the contacts of the native base have been dominated. So what we did, and sometimes people got confused. Like I said, protein folding, and I'm going to repeat this again, protein folding has two particular aspects. One of them is the one, can I find a potential that folds the protein? The other one is the physics behind it. Can we make all these assumptions I told you before, are they correct? And what I can do with that assumptions. Say basically, if I believe the protein is minimally frustrated, if I take that as a given, what can I learn? So what we decided on those days, the following thing. Let's play the inverse game. Let's look at the native state of the protein. Let's look at the native state of the protein. Let's write a glow potential, a structure-based potential like I told you. A potential I say, just native contact attractive and no native repulsive. And then I'm going to measure my phi values and everything. Now, I'm writing a model with no energetic frustration. I know that a priori. Right? So I cannot be caught on an energetic trap because the model doesn't allow for that. That's what I'm building into the model. If my results now show very good agreement with the experiment, that tells me that this assumption is not a bad assumption for the protein, that the energetic frustration is small. And again, I'm going to go back one slide just one more time. Notice that this is a good example. Look at the Go model and the three-letter code. They are more or less high phis and low phis. They have all the same contents. So it tells you that as long the energetic frustration is small, these two models are actually, you can learn a lot from this model. Doesn't mean, so even the experiments that, like my three-letter code, that this is what I get from my simulation, I can almost do good prediction about what's important, what's not important for me. So what Janeiro did, and I think she's going to tell you a lot about coarse-grained models and resolutions and stuff like that. This is the simplest model that we could have done. Right? And the amazing thing is, I really like this paper. That's the history of my life, is to write simple papers and get people 10 years later saying that it doesn't have all the details. Yes, but the paper said that. First thing we did. It was kind of fun to observe people. You cannot do everything with a C alpha goal model. Oh, thank you for telling me. So, but it's always good. And why I'm presenting this thing here, I expect you guys are smart and that do not make stupid statements like that. Because models are models. For some things they are good, for some things they are bad. There's no such a thing as perfect model, otherwise it will be a model, the real theory. Okay? If you work on theory, remember there are three different evolutions as you develop a theory. First, you start with an observation. An observation is something when you make a plot. You notice that if you vary a parameter, some other quantity varies. You can get it from experiments, and then you say, okay, observe that the degree of collapse is proportional to the energy. But that's an observation. You don't even know where it comes from, but you observe some correlation. You know the statistically significant. That's an observation. That's the first step. And then you make a comment. So say, oh, your energy depends on that particular variable. Sometimes you don't even know why, but you just write a relationship. That's an observation. The second one, based on those observations, you try to build a model that actually can mimic that observation. And the model is good. When that model becomes predictive and can explain and tells you always the limits of validity and stuff like that, and everything becomes to the nose, that becomes a theory. Right? If I give you two electrical charges here, and I ask you what's the force between them, you apply Coulomb's law, and you don't have to go and check on the lab to believe that's the force. That's a real theory. Rise to the level that basically you just apply that and you don't need to check in the lab. You know that's the answer basically. You have done enough to know that thing's predictive and we don't have to do it, but that's the last stage of these things. Okay, so it's very important that you have this element. So what I did with this model was to check this idea, if really the topological effects, the geometric effects are important. So what we did is, cartoon here, you pretty much got this protein here and you make like a C-alpha representation. It's the lower representation where you just put every residue as a bead. Now, you have interactions between these beads. Remember, when I did that, what my interaction have? This interaction is a very effective interaction. Because the interaction includes all the effect of solvation, plus all the effect of packing of the side chains. All these things are into that interaction. This effective interaction has also some geometry parts of it, because they're not only the side chains by part. So because of that, what we call this sort of a contact energy here, just look at the bottom one here, I'll go to the top ones. These things just tell you, when you have two things in contact, they attract, they're repulsive. This thing is real effective energy here, right? And as you are going to listen on coarse grain, you can ask, can we do better than that? In a sense, the basic, can we see how much of this effect comes from packing of the side chains? How much? But at this level, we cannot do that. So the model is just check the basic idea. There are 4 terms here into this potential. Let me tell you what they are. The first 2 are just to make sure the chain is for real, right? So the first term tells you these things just like a spring, keep the bonds in the right place. The second one is data on a C-alpha model. It just tries sort of to mimic the amino acid angle, try to make sure that these things are not, the angle between these 3 residues are kept. It's sort of a poor man's approach of, because you don't have all the C-alpha, C-beta, but you're required to do that. So these terms here, just make sure, on the entire simulation, fold or unfolded, these are, and this data change very little. They're just feel a little bit like harmonic coordinates. The other terms here are the important ones. This term here is dihedral. That's to mimic the secondary structure, right? So the dihedral angles is the dihedral angle between 4 residues. That's to mimic your secondary structure term. And this is to measure the contacts. Okay? This is the model. It's a very simplified model. And we just say the native dihedrals and the native contacts are attractive, and the non-native are repulsive. Now, still have 10 minutes, I'm fine. So what you have here is these 2 terms just keep your chain in contact. That's the simplest possible model. And these 2 terms here they are attractive. Here we have 10, 12 instead of 6, 12, because on the C-alpha model, you want to make sure that this residue is able to attract this one, but not that one. You want to make sure that you just attract nearest neighbor. Since these terms are very large, you won't be able to see this thing die fast. That's why you use the 10 instead of the 6 here. Right? So otherwise you have an artificial, but again, it could be anything. It could be a Gaussian. Make just things in contact. A particular shape here is not important, but just to raise the sugar. What's important is to make sure, like I told you guys yesterday, that you have contribution of both secondary structure and tertiary contacts. In a sense that you want the secondary structure and tertiary contacts form at the same time. If you have the secondary structure much weak, like I discussed yesterday, you have a sampling nightmare. If you have the secondary structure too strong, the same. So I think in our case we use what, about 30, 70 or 40, 60, right? More or less the stability, right? Between these things. But that's what happens. Now, I want to close showing you just a couple of results, it's sort of on our old paper, just to sort of bring these things to speed. And the results I want to show you here is in this plot. This plot here is the results of the simulations for 2 very simple proteins. And since you're going to be seeing lots of these things as we come along, let's use 2 cases just to introduce to you what they are. This protein is an SH3 domain. It's just like the end of the domain of SH3 that we cut out. And this is CI2, trypsin inhibitor 2, that's in here. Now, the people that have been working on that, particularly with structural biology, do quite well. You have these contact maps here. So forget about the colors for 1 second. Each box into this contact map here tells you that's a contact on the native structure, right? So it tells you that here residue third is making contact with residue 18. That's what the box means. Every contact on the native structure is in there. Okay? To facilitate your view here, there was something that Chile developed a long time ago. She sort of put on the side where the beta strands and the alpha helix are, so it's easier for you to look at the structure and map into the contacts. Just make your life easier. Start with a small piece of beta strands, then you go to this sort of here, then you go to the helices, then you have these 2 strands here, and then you have the final. It's easier to follow the structure and the things here. Okay. Let me tell you about CI2. If I look at CI2, I say, CI2 is a pretty damn good funnel. I say, hey, what do you mean it's a pretty damn good funnel? You told me that if you had a perfect funnel, you went to transition state, every residue should have the same probability. Now, you haven't told me, but here, I can see from your vary between 0 and 1. I know what the scholar map is telling you. If you go to that free energy profile into this problem, look at the barrier, you say, what's the probability of that contact to be formed on that barrier? And darker colors tells you you're very structured in the transition state. And lighter color says you're weakly structured. And I'm not seeing everything here in light red. I'm seeing some very dark ones around here, and I see some very white ones there. So what's going on here? It's because when I presented the lattice, I didn't put the full story behind this problem. That's the following situation. The reality is basically you have to add Flory entropy to the problem. In addition to everything else, you have to say contacts that are closer to the chain, they're easier to form because it's smaller loop to close than contacts that are very far on the chain. Right? There's a Flory entropy that comes from this log of the loop. And then it happens. But that's just telling you. It's just, the story is just like I told you. Every contact counts the same, with a little hierarchy to say the contacts that are closer are formed first and the other next after. And that. So if you add the story, every contact is the same, correct for the hierarchy, that ones that are closer on the chain comes first and the other ones later on the chain, you see these things give you the right answer. Because see, you're darker in diagonal. Means when the contacts are very close to the chain, as you move away from diagonal, you get lighter and lighter. So it tells you that you don't have any, it appears to be very funnel-like. It's just like the same perfect funnel I told you, with just the difference that shorter contacts are formed first and then longer and then the other ones. It's great. Tells CI2 is a two-fold protein. And if you look at the experimental paper, people say, oh, the transition state of CI2, the alpha helix is formed, and the mini core is formed. It's exactly those contacts here. So we agree exactly with what the experiments predict. But it's not only we that agree. Everybody agrees. It's no credit to simulation that we agree with the results here. It's a credit to the protein. You have to do something pathetically idiot to get the wrong answer to this protein. It's such a robust folder. It's such a good funnel. It's such a thing that basically is very hard to get a different answer. It's very hard not to agree with these experiments here. It's a robust, good folder. Tolerate a lot of abuse CI2. The other proteins, these things are not so clear. But this one, you see, every theory agrees. And like I said, no credit to theories, credit to the protein. She's made to make theoreticians happy. Okay? That's this protein CI2. So, since I'm running out of time, but I have to go through everything today. This is a typical case where the funnel situation is not so great. And the way you see that is because you have a few contacts very far from the diagonal that have been formed early. That's a major inconvenience. There's so much proteins can tolerate on that. And that tells you this is not a perfect funnel. It's not like you can come by all possible paths. Some contacts have been formed early. That creates a certain level of difficulty. Makes harder to fold. But that's the fact. And you know this is a geometrical fact. That's not an energetic effect. That's what we call that's a fact that comes from the geometry. And here's my last slide for today. Shows you SA3 and tells you that base SA3 in terms of the fold. Here's a cartoon of the folding of SA3. So here is base one of the two hairpins that get being formed. And you see that first you form all this local structure, these local hairpins, like I said, the short range contacts. But then, in order to fold, you have to get this distal hairpin and bring together with this diverging term. You have to bring these two parts together before I can close the core. And if I don't do that, I don't fold. And that's a problem for this protein that creates long term. So I want to close here to say, great, that's a geometrical aspect. But there's a beauty that comes from this figure here. That tells you, no, basically, how do we know that's not an energetic effect? If I can put an explanation just based on the native structure, then it's not an energetic effect. If I'm right, if I just use the native structure, it tells me the native contacts are dominating over the other contacts. You may have a geometrical feature here, but the geometrical feature is the order of contact formation is important. It doesn't mean that you have no native contacts creating traps in the situation. Okay, since I'm getting late, I'm going to stop here. But I hope I gave people a good feeling that when you listen to the fall next lectures and things like that, what simulations are all about, what are the sort of the difficulties that you're going to have in life as you're computing these things. And at the same time, I think it's very important that if you run simulations and stuff like that to behave as a professional, okay? It's the same when you do experiment. The same way you should not run an experiment if you don't know what the heck you're doing there. You shouldn't run a simulation if you don't know in heck what you're doing there, because your chances of making stupid mistakes increase enormously if you don't know what you're doing. Okay, thank you.
Maybe
We're late, but I'm going to take a couple questions.
Yeah, we started a little bit late also.
Why does higher probability mean that it falls faster? Why is that? Why the last slide you said it was higher probability that the contact formed earlier?
Okay. What's telling you, that's a very good question, and I'm glad you asked. Here you're just looking at the contact and transition state, and I'm extrapolating. So basically, let's be honest here. I gave you a little bit of a under the rug situation because we're running out of time. Here, all I'm looking at is I'm looking at my free energy profile, like I showed before. I let the transition state and say which contacts are formed there. All I have from here is a static view of the transition state. And I'm trying to give you an order of folding events based on the static view. They work, but basically, all it tells me is at the transition state, this part is well formed, this is partially formed, and this is not formed. Now, that's a good indication that probably things are forming into disorder. This one tells you the following thing. That at the transition state, yes, you have the state schools diagonal formed. But you have this long-range contacts formed. When you have much contacts, they're much shorter on the chain. They're not formed yet. So that stage tells you that you have short-range contacts. Contacts are short in a distance, shorter on the sequence. They're not formed yet. So what I'm telling you is, in the fold of this protein, here, at least that you can affirm. You have to form this part before you form these other parts. And that's a requirement here. As I'm going to show on some proteins, sometimes this is required, but the protein doesn't know. And what makes is make these contacts too early, and then what you have what you call a geometrical trap. Because then you have to break these contacts and form the long-range one before you do. Situations like that can create traps for you. Okay, but that's what's there. Go ahead. 1, 2.
Do you think that frustration can be designed in proteins? Do you think that frustration can be designed in proteins in order to guide the protein past geometric barriers?
That's a very, very good question, okay? And I'm going to tell you a couple of things. What you're asking is something that's a very active topic of research. Do I believe that energetic frustration is always a problem, or can I use it for some productive things? I think Daniel is telling me a little of networks this afternoon again. He's going to say, do you believe that noise is always an annoyance, or noise is required to compute? These are interesting questions that come to it. My answer to you is, on this initial folding problems, we chose these well-defined small proteins where you're not supposed to do anything too interesting, at least at this level, so we don't run into these problems you have. When you start to go to larger systems of proteins, then you start to see situations where you may have to do that. So the question is, can I sometimes create some energetic traps that avoid me to fall on the wrong geometrical hole? I haven't seen this yet, but it appears to be a good thing you can do in the future. What I've been seeing is proteins are required to have multiple states. They have to have competing interactions. So proteins supposed to bind here to bind there. They have two shapes, allosteric and stuff like that. Then these proteins may have some frustrating interactions because that's a way to say, if this one wins, I'm on this shape, that one wins, I'm on the other shape. So you start to see that basically sometimes loops, they're supposed to bind to DNA, they're very disordered in the absence of DNA. Because just when you put the charge interaction on DNA, you can actually optimize the energy. And you want that thing to be just flexible and shaky before that, before DNA forces it to go to the right shape. So, but if you look at protein, just on its way out, it's not like that. So this falls in lots of traps that move from people call them intrinsically disorder or stuff like that. That the fact is that not in every case protein is supposed to go and form rock solid. So in this case, you're going to see that their things are in there. Peter, if you look at Peter Wallman's recent papers, he has been very interesting. He's doing an exercise here. It's a very dangerous exercise, but it's a very interesting exercise. Why it's a very dangerous exercise? He's trying to use his structural prediction model, and he's trying to figure out which are the frustrated regions of a protein and which are not the frustrated regions of the protein. Why this exercise is dangerous? Because that means that you believe your model, not only your model is good enough to predict the entire structure of the protein, but it's good enough to give information about small pieces of the protein. It means you believe your potential is damn too good. And so, but taking that aside, we can argue for a lifetime the potentials are good enough to do that. Right? So it's a very dangerous game because of that. So you can start to observe why some areas of protein are required to be frustrated. And then when you observe a frustrated area, do you think this is just leftover the proteins can deal with, or leftover from evolution? Or this is something that actually, this frustration is a sign they look the protein alone, and that's the area that's going to interact with another folding partner, or has another binding protein, or is there for functional reasons? I may tell you a little bit about that in my last lecture. Right, but that's the kind of directions that we may want to go in terms of frustration. I didn't have time to go there, I tried to stay here, but that's the direction to go, right? It's basically, when you try to believe that the landscape for a protein is not only for folding, it's folding and function, then you can start to put all this creativity into it.
I think you already said it, but I missed it. But in the Go-leg potential, why is it a 10 power?
Because that's a good one, because it makes no difference in details. But the bottom line is, these things are very far apart. These are potential 7 angstrom apart, right? So if I look at unfolded basin, and let's assume the native basin, I have residue 1 and 10 are attractive to each other. Now I look at unfold configuration. I have an unfold configuration where the protein goes around here, there's another residue here, and here. And this is 1 and this is residue 10. That's unfold configuration. If I use a 612, the 7 angstrom, means the range goes up to 14 angstrom. Then what you have is an attractive interaction between 1 and 10 here. That's non-physical. That's not real. There's a residue in between. But because you're using the Lennard-Jones form, you know what to do, because you have the same going all the way up to 14 angstrom. It's too far. There's no problem in the native state, but it's a big problem here because you start to have attraction. So what you want to do is to make sure this thing die faster, so that you only have interaction just with nearest neighbors. And the Lennard-Jones on that potential is just an artificial thing. You could have a Gaussian, you could have anything to attract, but you want to make sure that's just short range. So it's just technical. Actually, it's not like you're that smart when you first run this potential. We did it with a 612 because everybody's lazy, just do whatever is easier. And then we start to observe very ugly structure unfold basin. What the heck's going up here? See, and then we switch to 1012. Then you sound smart when you present. See, you cannot do that because you have this problem. That's always you invert history, right? And it makes you sound like you're smarter than you really are. But that's the reason.
Yeah, José, I think the comparison between these two programs are really nice, but I think it would be even greater if you could, for example, for CA2, in Alan First, they made a fragment that the protein, although he said, well, the structure is the same, but we found, by making experiments that was much less stable. That was the fragmented protein, so just breaking to pieces. And
yes.
So that would be quite interesting if you could do. I mean, of course, you have.
I'll let Cecilia show you that to you.
Ah, she has done that?
She has done that.
With the fragment? Oh, you have done it with the mutation, right? Yeah. With the fragment, right? Because you have the true fragment of CA2. Oh, so that would be quite great.
I'll let you show to you.
Yeah.
It's her work. She knows better than I do.
Okay. Okay,
But that's
so I'll wait.
all these things can be done. Right, you are absolutely correct on these things. And there are lots of, actually, we haven't had time to go in there, and I think we are going to try to a little bit of this stage later on. Is that based on this question, the other questions, what happens when you start to have a little frustration? Is this thing stable enough? How things go? I think now we're in a good position to do that. I think Gigi is going to tell you about that, how we play these games on real games. We feel that now, at least with this crowd, we have everybody on the right stage to go there, but that's a good point. Daniel,
Yes,
are we happy?
we're happy.
If anybody wants coffee? Okay.
That'll make people happier.
And sorry, it was also go to the secretary for Thursday night,
Yeah.
and then the Saturday session is
And this space for Friday.
okay. Okay. And it's my fault that the poster session was put later. I was sort of thinking Gordon conference rules, not karaoke rules. Okay. So in today's lecture, I want to discuss mainly some issues about organizing principles. I want to finish up some on kinetics as relevant to protein aggregation. Then I want to talk about organizing principles for amyloid structure and the extent to which those connect to amyloid toxicity. And so I will hit two organizing principles of amyloid toxicity today and three organizing principles of amyloid structure today. That's my plan. So where I kind of left off yesterday, I thought I would pick back up again to talk about the prions. That's the area of amyloid science that I've worked the most on. And I didn't get a chance to go through this, but it helps to remind you why the prions are so interesting and remain so exciting to people. Oops. It's on, but it's not. Oh, there we go. I got it. There we go. Let's see. So the prions, as I mentioned, are alone among all diseases. It could be inherited, spontaneous, or infectious. Why are they hypothesized to be protein only? Basically because they're pretty much indestructible compared to other diseases. You have to heat them up to the order of 600 degrees Celsius to kill them off, so to speak. You have to, they're resistant to UV damage. They're resistant to proteinase damage. Proteinases being enzymes that will consume other proteins. And they're resistant to a lot of the things that will basically destroy bacteria and viruses. It's also known that when you take this particular protein, the PRP protein gene, and you knock it out from mice, those mice will not get infected, but those mice also, for some reason, again, related to the lack of knowledge about the function of prions, nothing happens to the mice as far as we can tell. I don't think I want my PRP genes knocked out just to play it safe, but as far as we can tell from the mice, whatever happens to them is very subtle. I talked yesterday in the discussion about this test tube business, but there have been some substantial efforts to try to synthesize prions de novo, synthesize them in the test tube and cause infection. And I think we're getting close to the point where viruses are certainly ruled out by the Sapodopome data, and we're getting close to the point where people are at almost 100% acceptance of the prion hypothesis. I mentioned the idea of strains yesterday, that there are these different strains of prion diseases, and the strong belief is that strain is encoded in conformation. When I talk about the organizing principles, I'll mention a couple of ways that could happen with strains. They show up with different incubation times, different distributions of the spongiform lesions where there's actually neurons deleted from your brain, different numbers of sugars attached to the prion protein. And there is this aspect of exponential growth, and it appears that the minimal infectious unit is about 3 to 6 monomers based upon biophysical studies. And the same study indicated that the most effective aggregate size was about 20 monomers or so of prion protein. as we discussed a little bit yesterday. And I mentioned the yeast prions yesterday. I'll just flash this slide one more time. But again, one of the fascinating things is that the prions, they're not mammalian prion in sequence, but these prion proteins in yeast and other fungi actually serve a functional purpose. And they do have the exponential growth, and the strain is in the conformation, as these AFM images from Weissman's lab were discussed yesterday. Okay, so one of the things about the prion protein is that, I should have perhaps have a picture of the prion protein, but at the C-terminus of the prion protein, there is a so-called GPI anchor, which means that when you send the prion protein out from the cell nucleus to the surface of cells, and there's really only two kinds of cells in the body where it gets preferentially expressed. One is neurons, and hence all the damage to the nervous system. The other is a particular cell in the lymph system, follicular dendritic cells, where it doesn't seem to cause damage, but it does replicate. So it is a problem when it goes to these follicular dendritic cells. In each case, the prion sits on the surface of the cell. So there's this sort of random part, and then there's the structured part, and then there's a GPI anchor that sits right down into the lipid bilayer. It intercalates right down into the lipid bilayer. Okay, so the prions are membrane bound, and in fact, one of the issues for these diseases when you talk about mechanisms for how they can kill you, one of the issues for these diseases is to separate which part of the cell they belong to. There could be different mechanisms involved in different parts of the cell and not one universal mechanism for everything. So since the prions are membrane bound, instead of dealing with these one-dimensional kinetics models, it's interesting to ask about whether there could be some differences if they aggregated in a two-dimensional way on the surface of the cells. And that's further motivated by the discovery a few years back of two-dimensional crystals of infectious prion protein. This is an electron microscope image with about 12 angstroms resolution taken by Holger Willi in the Prusner lab at UC San Francisco. So this is you notice that there's a beautiful hexagonal faceting to this two-dimensional crystal. It really is, with signal averaging, a two-dimensional hexagonal array. And each one of these little donut-looking things is one of these, presumably oligomers of prion protein. So there's already evidence that you can form infectious prion material into crystals, not just the fibril structures. And so one of the things we did was to ask about whether two-dimensional kinetics might give you something interesting. And we took some very simple models, and it's not bad to do these simple kinds of models. In this case, for somewhat similar reasons to what Jose said, just to see what you can learn with simple models that might be transferable to more complex models. One of the questions is whether, and we've already talked about this some, whether you could actually have this autocatalysis at the monomer level, where PRP could come in coupled to PRP scrapies, and then you could have two infectious PRP scrapies, or whether you have to have a picture like this, where you only convert to this form upon attachment. And we sort of, as a misnomer, call that autocatalytic aggregation, meaning that you convert to this form, so this one does go to this form. The reason it's not autocatalysis in the same sense as this is that this growth process won't per se give you exponential behavior, whereas this will give you exponential behavior. You know, you have two copies here with one copy on the way in. You're clearly going to get exponential growth out of that. What aggregate structures and sizes best correspond to experiment? Do aggregate shapes and sizes influence fission? Because if you're going to get exponential growth out of this, you're probably going to have to break it somehow. And can you reconcile the timescales for infectious and sporadic diseases with a model like this? So I wanted to show you a few images from some work that we did a few years back. I'm pretty sure I included this paper in my packet over here. So this was done with cellular automata simulations and some analytic theory. So the green ones are normal prion proteins running around on the surface of a model nerve cell. This is a seed, an initial seed placed in there. We also did simulations where the prions were allowed to diffuse around on the cell. What we gave them was we gave them rules for interaction. You could describe it with potentials. We decided it'd be easier to use the cellular automata rules in which potentials are implicitly encoded. And then after time, you get a larger aggregate, and it turns out that you get a considerable acceleration of the growth process, partly because of the larger surface area, and partly because of the nature of the particular growth process that emerges in this simulation. So if nothing else, this proves you that my graduate students can make movies. And so the idea is basically this, that we let these prions diffuse around on a two-dimensional surface. That would be the normal prion. We can put in these seeds, and if we make a microscope of where these attach to the seeds, then we give it a coordination rule. Okay, so the idea is this, that we have sort of an effective free energy surface if the PRP comes in and it has, so the squares are the bad guys and the circles are the good guys, also enunciated by G and B. If the prion comes in and there's one or there's two, the prion comes in and there's one other of the bad guys, then it's going to always be preferential to be in the good state versus the bad state if you draw an energy diagram. Okay, so if you have one or if you have two of them, it's always going to be in the good state versus the bad state. But there's a cooperative modification of the energy landscape, so that if you go to a situation where you have one good and two bad, then you give it a 50-50 probability of being good or bad. And if you go to a situation where you have one good and three bad, which we call critical coordination 3, then we always make it favorable to convert to the bad form. So these are simple rules. There's no energetics put in this. It's just simple rules that you're going to convert to the bad form if you surround it by enough bad guys. And if you don't have enough bad guys, you're going to stay in the good form. Okay? And you can tune this critical coordination. You can make the critical coordination 1, 2, 3, 4, whatever you want. It's a tunable parameter of the model. So the parameters in the model basically are the concentration of the normal prion, the number of seeds that you put in, and also these coordination rules here, the critical coordination number. So it's a very simple thing to simulate on the computer. And nonetheless, it gives you some interesting results. So, and the model we had in mind was the following, that we would basically put in a seed, we'd let it aggregate to a critical size, and then we would imagine that some process would come in and that thing would fission, it would split. You can ask, why would it tend to split? Well, if you go back to this picture of the two-dimensional aggregates back here, these aggregates are going to want to be flat, but there could be processes on the cell membrane, either as they come close to the edge of a synapse, for example, where there's a lot of curvature, there's membrane undulations, or the other thing is that the prions are sent out in vesicles and they come back in vesicles. So there are these little caveolae which have pretty high radius of curvature. If one of these flat fragments comes close to that, it's going to tend to break the fragment, given that the prion is also anchored to the membrane surface. So one aspect is that there's a competition between the tendency of the membrane to curve and undulate and the tendency of these aggregates to want to be flat, which could lead to breakage. Honestly, if there is breakage, we don't know what the mechanism is, but we postulated that there was, and therefore that we would look to see it get to a size that we thought would be characteristic of breakage. The actual size there it's not too sensitive to which size you pick, as long as it's basically nanoscale. And then the breakage would take place, and that the incubation time distribution then would be dominated in shape by the time it takes to get from the seed to the critical aggregate size. We did that for 5 different concentrations. That are these curves right here. And this is the incubation time distribution, or sorry, the aggregation time distribution, the time it takes to get to this critical aggregate size, scaled by the peak, scaled by the mode of the aggregation time, or I'm sorry, scaled by the median of the aggregation time distribution. Now, if you do these 5 different concentrations, they all just collapse on top of one another. So you're seeing the same phenomena for all of the different concentrations. And we were quite pleased when we noticed that there was some, not experimental data, but there was the following available. As you know, a lot of cows died in England from mad cow disease. In fact, hundreds of thousands of cows died in England. As a result, there was substantial epidemiological data, and the epidemiologists separately went after to try to figure out what the incubation time distribution was for cattle in England. In other words, what was the distribution of times that these cattle got sick from the disease? And the peak in that incubation time distribution, if I were to put it on here, is about 5 years. That gives you a time scale for cattle. These points are taking the best fit to that incubation time distribution. In other words, a separate calculation, if you will, but a calculation used to fit the data for mad cow disease. We just scaled it by the peak position, and we could plop it right on top of our curve. We didn't do the data to generate the curve, but it happened to work. It doesn't prove anything about our data being correct, but it does suggest that the possibility is there that if you have a low number of these seeds that lead to these critical aggregate sizes, that is a hypothesis consistent with giving you this very fluctuation-dominated incubation time distribution. And I say it's fluctuation-dominated because if you look from the starting time, the width of the distribution is comparable to the peak of the distribution. And so in other words, there's a lot of fluctuations in time to reach these nanoscale aggregate sizes, and therefore you're going to run into a very broad distribution. Now, the one thing you can ask in comparing these is the cattle. Does anybody know about how the cattle got sick in England?
Late 8th.
They ate this tainted protein supplement. And as they say, they sort of made the cattle virtual cannibals because they fed cattle into the rendering plants to make protein supplement, and that's how they amplified the prion in the process.
It was from the cat itself.
Yeah, that's right. Initially, it was probably some animal, some roadkill or something that was tossed into the rendering plant. In any case, then they got fed this tainted protein supplement, and if you ask, there's probably very few of the prions that survive the rendering process. But enough of them that they get into the gut, but then only a few of them from the gut would get to the central nervous system. So you're definitely going to be to this limit of dilute concentration of prions in the process. The one thing I wanted to mention is that you can, the one thing I want to mention about that curve there is that you can actually get at it analytically. I'm not going to give you all the details, but you can get at that curve analytically by doing what's called a waiting time or arrival time distribution. And that's discussed a little bit in the article by Ferrone, which is in here, and it's also discussed in the article by Kulkarni et al., which is in there. But basically, you can say, if you want to attach in monomers with attachment probability p sub j, and that attachment probability would be just a constant if it's simple growth, or if it's the branching or heterogeneous catalysis, you might have something of this form, heterogeneous autocatalysis. So if it were the branching or the nucleation that I talked about yesterday, you would want to have a growth rate which was proportional to the number which you were attaching, j. Then you can define the joint probability distribution for those attachments per unit time would be equal to p sub the product over j, p sub j e to the minus pj t, and the arrival time distribution p of t would be equal to the integral from 0 to infinity dt1, the integral from 0 to infinity dtn, p of t1 tn, with a constraint. And this delta function, if you're not familiar with delta functions, all it tells you is that this thing is basically infinite when you satisfy this identity inside the delta function, and it's 0 otherwise. Okay, so so subject to this constraint, you take this joint probability, and this gives you the distribution of the arrival times. And then it turns out for these two different cases, you can work it out analytically. I won't go through those details here. But in case one, it gives you a so-called gamma distribution, where p of t has the form pt to the n minus 1 over n minus 1 factorial e to the minus pt. So it depends, it's very flat at the beginning, and then it will have an exponential tail. And so we'll give you a shape similar to what we observed in our simulations on the prion problem. And two, if you have this case, too, then p of t I have to actually check my notes on this one because I never remember this form. It's a sub n e to the minus p plus p prime times t times 1 minus e to the minus p prime t raised to the n power. And that also will come in, because of this subtraction here, that will come in very flat and have this, this peak in it, like that. And it turns out that for the simple two-dimensional model that we considered in these simulations right here, that this describes the data very well. And you can motivate it based on the calculations. So now I want to just show you that you can learn something else if you just play around with the parameters of this model. So this is a situation here where we let the critical coordination be 1, meaning that if you had a dimer of prions, that would suddenly be stable in the aggregate form. And the next one is when we looked at critical coordination 3, with a single seed, I should say. The next one is when we looked at critical coordination, sorry, this is critical coordination 1, critical coordination 2, and this is critical coordination 3. And the stable nucleus is more complicated for critical coordination 3, and it has 10 monomers in it. So in other words, here you could put in a dimer, it would be stable. Here you could put in a trimer, it would be stable. Here you have to put in something much larger. Now what's interesting is look at the timescales here. This is for a particular low concentration on the surface of our model cells. This one is seeded, this one is unseeded. These, it turns out that it doesn't matter whether you seed them or not. The process of growth is so rapid that if you put in a seed, it helps a little bit, but otherwise you've got these dimers and trimers popping up all over the place anyway. So the significance of that is if this were the right model for prion disease, we'd all be dead. Okay? Because it wouldn't matter whether you seeded it or whether you started out early in life with a little bit of aggregate. It grows so quickly, we'd all be dead. Notice also that the width on this log scale is about the same for these 3 distributions, which means if you put it on a linear scale, this one is much broader. It makes sense. It's got to be much harder to get to this critical nucleus than it is to just get to a dimer or a trimer. Finally, let's say that this is a reasonable description of the incubation time distribution for prion diseases. Does it give us any insight into the timescales? We talked about how to get slow timescales for the poly-Q diseases with a much simpler argument. Can we use this more complex argument to understand something about the prion diseases? Well, here's the idea, which was already hinted at by the kinetic control model yesterday. That disease of ritual cannibalism in New Guinea has an incubation time of about 12 years. Now, if you scale from that to this incubation time, where we did it with no seed at all present, we just let the stuff eventually aggregate, that would go from 12 years to something like a 1,000-year peak right here. There's a little tail to this distribution coming down in here. And again, the 1 in a million would be sort of finding out that little tail down in the human time window down here. So the upshot of these little calculations is that it can reconcile the difference between infectious prion disease and spontaneous prion disease in principle. It doesn't prove that this is the right answer. It just suggests that should we prolong the lifespan to 1,000 years, there's some more public health threats lurking out there. Okay, now getting to application of this kinetic theory in another context, what is the mechanism for exponential growth? And it turns out that there are experiments which show that the membrane has to play a role. And these are pretty interesting experiments. They just appeared a couple of years ago when I was on sabbatical at San Diego. What they found out was they engineered mice such that, remember I told you that the prion is stuck to the membrane with this anchor. So here's the prion protein, and it has this anchor that sticks it into the cell membrane. And that anchor attaches at the C-terminus to a couple of residues at the C-terminus of the prion protein. So what they did was they engineered these mice such that this region where the anchor attaches is just gone. And so then when you express the prion protein, it's kind of like untethered astronauts. It goes out to the surface and it goes out into the free intracellular space and just floats away from the cell. Then they found and they found good evidence that that's exactly where it went when they looked at the mice after they passed away. Then they would inoculate the mice with a particular lethal dose of the PRP scrapies, for which wild-type mice would get symptoms at about 100 days, I think. And then they measured the amount of infectious material that grew in these mice's brains, and that's this curve right here. This is where the wild-type mice prion level would sit after the mice had died. And if you put a curve of the same dose for the wild-type mice, it looked like this. You come out here and boom, there's this exponential growth, and boom, then they saturate it out, and of course the mice die somewhere right in here. These mice didn't die or get symptoms out to 600 days. So that tells you that the toxicity mechanism is connected to the membrane. The toxicity mechanism is connected to the membrane. But notice also, I'll ask the same question I asked yesterday. What's the simplest function that could describe a shape like that? Simplest function of time that could describe a shape like that. They have Jeopardy back here in Brianna? Quadratic, thank you. Quadratic. Okay. A quadratic in time describes that. And so in fact, if you take this and you plot it versus t squared, you get a beautiful fit with a regression coefficient of about 0.97. That's t squared. So not only is the membrane critical for toxicity, it's critical for the replication of the prions. It's critical for exponential growth. So you learn something else from these experiments. And in fact, when we use the kinetic analysis, that t squared analysis that I talked about yesterday, you can apply that to this problem to try to make an estimate for the ratio of the PRPC that goes in between the cells to the ratio of the PRPC in the wild type. And you get about 30 times more PRPC out in the intracellular medium than you have for the wild type PRPC. And you might ask, well, why is that? Because the PRPC gets recycled. The PRPC goes out to the surface of the cells, and then it comes back into the cells. And that may be crucial for the toxicity and for the exponential growth. If you happen to grow this stuff out in the intracellular medium, so in the intracellular medium, if you grow these aggregates, which are these brown things, no big problem. Doesn't hurt the animal, at least out to 600 days. Once you get past 600 days, there's so much amyloid present in the poor mice that they wind up getting it in their heart, and it kills them. Okay, but this is another example of how you can take this simple kinetic analysis and actually learn something from the data. So if you take nothing away from these talks of mine, I hope you take away that there's some very beautiful and simple kinetic phenomenology that can be applied in a lot of contexts. And I'll remind you that you can read about it in the paper by Ferrone, which is in this packet right here. It's very simple, it's very pretty kinetic analysis, and you can apply it in a lot of different contexts. Okay, now, this was sort of drawn from a talk that I called Amyloids, the Good, the Bad, and the Beautiful, which I think I subjected Jose to. But, and the beautiful, I think, about amyloids, we've already heard about the bad. The bad is that it kills people and things. The good is that they can be useful for some organisms, like yeast, right? And the beautiful, I think, is the organizing principles, because there's something really pretty about the growth of this amyloid matter. And trying to explain how you can get this spectrum of amyloid structures remains an outstanding problem in biological physics, biological science and biological physics. So one of the organizing principles which clearly works, you can grow amyloid fibrils this way in the test tube, and it may have something to do with some of the amyloid diseases as well, is to extend the minimal frustration concept that we heard so elegantly explained by Jose in his lectures, to extend the minimal frustration concept by the concept of domain swapping. So in domain swapping, what you do is you consider, for example, a protein like this, which is the human cystatin protein. It already has quite a bit of beta sheet structure in addition to this alpha helix. And then what you do is you take some of the domains from one protein and you hook them up to the same contacts, but on the other protein. So in this dimer, we take this green rung and the alpha helix from this side and put it over here and attach it to the blue side in the same way as we would have attached it in the monomer up here. Similarly, we take the alpha helix and this stretch of beta strand from the blue side and attach it over here to the green side. It glues the things together. Clearly, energetically, it doesn't cost you any difference energetically between these two. Basically, what it will, of course, cost you is some positional entropy. But if you go to high enough concentrations, you should definitely favor this kind of thing. And there's a couple of nice papers. I'm advertising for Jose and his group here, but there's a couple of nice papers that were done about this using these kinds of Go models that Jose has already talked about and we'll know more about from Cecilia. For example, there's this SH3 domain that we already saw shown today, so I don't have to say a lot about that. This shows a contact map for this red one on its native structure. Here's a contact map for the blue one. And then this shows the domain swapped contact map for the SH3, where now we take blue and red and we line up the native contacts as if though they were the same native contacts on the red or on the blue. And so that's the notion of this domain swap. And so Xichen Yang and Sam Cho, along with a number of other authors in this paper, did these Go model simulations on this, where they used the contact map to make a symmetrized Go model to describe two proteins interacting. And in order to make sure that those proteins would sit in the same vicinity, they used a harmonic potential, sort of like what they use on the Bose-Einstein condensates in physics. They used a harmonic potential to get the monomers close together to allow them to potentially domain swap. And then you can actually try to remove the effects of the harmonic potential. But this paper showed some free energy surfaces, which are quite nice. So this is in units of the folding temperature. This is above the folding temperature here. This is just below the folding temperature here, and this is well below the folding temperature. So this is where you have two isolated monomers, and you have a deep free energy basin, which is marked by blue on this profile. The order parameters here, these are intra-chain contacts, that is monomer to monomer contacts, and then these are interchain contacts between the two monomers. So here you get a deep minimum around the dimer, and you also have a deep minimum around the two monomers. And then as you go up towards temperature, you see these interesting intermediate states forming. So the Go models, even though you might think that you've got the answers built into them, they actually can teach you interesting things about the kinetics and the stages of getting from unfolded to folded. And you can see that there's these two different pathways. You can either go off this way towards the dimer, or you can go off this way towards the two monomers. And then as you get the unfolded random coil structure over here. So these are interesting things. So the principle of minimal frustration can be used to describe this domain swapping, and this domain swapping clearly has been shown experimentally to allow you to grow amyloid fibrils. Another work that was done by Xichen as part of his dissertation was to try to calculate, we already heard about potential amine force, so I feel like I'm set up nicely by Jose here. Xichen calculated a potential amine force as a function of the separation, and he found when you're below the folding temperature that there is a monomer and a dimer minima in this potential amine force. And then to get an interesting idea about the possibility of dimerization as a function of concentration, after removing the effect of the constraint, which you can do by sort of inverting the, in a way it's like doing an inverse Laplace transform to get rid of that harmonic constraint, then you can get this potential amine force, which is the free energy at separation R minus that out at infinity, and put it into a calculation of the second virial coefficient. And just to remind you,